Good morning, everyone. We're so excited that you're here. I'm Sharon Loftspring, and I serve on the board of NF Central Plains and the NF Network. When my daughter was diagnosed with NF1 over 20 years ago, I was devastated. But that dark cloud has turned into something much brighter. I've met people from all over the country that I now call my friends. I've seen how being an advocate can advance research, and most importantly, I've watched my daughter learn to overcome her challenges and grow into an amazing young woman. And finding that silver lining happened with this day of information. When NF Central Plains realized that we would have to cancel our in-person event this past April, we were so disappointed. But that dark cloud has also turned into something much brighter. Thanks to our partnership with the NF Network, we were able to bring these outstanding speakers to a broader audience. Those of you who are here today from all over the country, thank you for joining us. And a big thank you goes to the volunteers with a capital V who run NF Central Plains and help make this event happen. Nicole, Kami, Jill and Shelley, Jeannie, Janina, Alana, Rachel, Linda, Patty, Yvonne, and Peter. They deserve much more than a virtual round of applause. And now I'd like to turn it over to Brianna Brunowski-Stutzman, the NF Network's Marketing and Event Coordinator and my personal technology angel, who will explain how today's program will work.
Thank you, Sharon, and thank you, NF Central Plains, for putting on such an incredible event today. I am excited to get started. As a reminder, each of our speakers' sessions today are being hosted in their own individual webinars. If you haven't already, please be sure to register for the sessions that you are interested in attending. The links can be found in the chat box now, if I can get this in there. If you are unable to make each of the sessions but are still interested in the topics and the speakers that are presenting, please be sure to register anyway, as each of the individual webinars will have a webinar recording sent out to you within 24 hours of the webinar's completion. Let's see if this works now. Okay, I'll get that up in a moment. Our presenters are excited to share their wealth of knowledge with the NF community. Please note that our speakers are here to answer your general questions about neurofibromatosis during their presentations. The NF Network and NF Central Plains advise anyone with specific medical questions regarding their NF diagnosis to direct them to their NF specialist or physician. Any questions you'd like to ask the panelists can be submitted via the Q&A button found at the bottom of your screens. While we will be monitoring the chat, the easiest way to ensure all questions are answered is to submit them via the Q&A. We hope to have the opportunity to answer all of your questions. However, in the event that we do not get to your question, we will be sure to reach out today and we'll do our best to get your questions answered. The NF Network is proud to co-host this event alongside NF Central Plains. Over the years, NF Central Plains and the NF Network have provided essential resources and programs to our NF community through many avenues, including events like this. The NF Network's flagship program is the work we do out in Washington, D.C. The NF Network Advocacy Program and the NF Network have played a vital role in securing over $360 million for NF research. Those research funds made a crucial impact in producing the first ever treatment for neurofibromatosis, selumetinib. NF Central Plains joins us each year in DC, educating Congress on the importance of federal funding for NF. Here to talk a little bit more about NF advocacy, what it means to them, and how you can get involved is the NF Central Plains advocacy team.
In 2019, 80+ advocates from around the country, organized through the NF Network's advocacy program, raised our voices in Washington, D.C. for NF research funds. NF Central Plains had an outstanding team of 5, and I'm proud to introduce them to you today. I'm Sharon, and I represented those of you from Kansas. I advocate because I want to educate our leaders about NF.
I'm Jill, and I represented those of you from Kansas, Western Missouri. I advocate for my daughter, Riley.
I'm Rachel. I represented those of you from Nebraska. I advocate because my son has NF, and these federal dollars gives hope to all living with NF.
I'm Patty, and I represented those of you from Oklahoma. I advocate to help spread awareness of NF and I do this for my grandson.
Hi, I'm Ilana, and I advocate on behalf of those of you from Colorado. I advocate because I have NF and I want to put an end to this condition.
And I'm Yvonne. I represented those of you from Kansas and Tennessee. Although I don't have a personal connection to NF, my NF story is yours. And I'm happy to take your stories to your representatives in DC.
Not everyone can travel to Washington to advocate.
But everyone can participate.
Write your personal story.
Well, share it with your representatives.
Urge your congressmen and women and your senators to support NF.
It's easy. Watch for emails. We'll tell you how.
So speak up.
Your voice counts.
Research dollars turned into treatments and a cure.
That's what we're all fighting for.
Perfect. And Kim.
Oh, wonderful job. Wonderful job. I would really like to say I'm Kim Bischoff, the Executive Director of the Neurofibromatosis Network, and I also have a daughter, Jennifer, who's 37, that has NF1. We've been involved in this organization for the past 30 plus years. But first, I would like to say a huge thank you. Let's do that virtual applause to Sharon Osprey for putting this together for us. Thank you, thank you. And that video that you just did from Central Plains was gorgeous. Thank you very much. And also to our technical guru up there, to Brianna, thank you very much. Wouldn't be possible without you. Now I'd like to tell you just a little bit of a background about the NF Network. It was founded in 1988. It's an umbrella organization to facilitate the collaboration of national activities and networking of shared resources. NF Central Plains is one of our member organizations. We're built on two pillars. We're leading NF advocacy and we're building NF community. In leading NF advocacy, what we're doing is we're promoting NF research. We are the leading national program advocating for federal funds for NF research. We began this program in 1996 and have advocated, like Brianna said, for over $320 million federal dollars, which has already been allocated into NF research. We actively participate in scientific and medical conferences. We provide seed grants to researchers, and we educate our NF community about the possibility of participation in clinical trials. Our second pillar is building NF community. Through that, we use educational meetings like we have going on here today, webinars. We do this also in electronic and print materials. We do peer counseling. People call the office and we connect them with others that have NF. We do social gatherings and NF chats around the country. We do referrals to NF clinics so individuals get to the proper medical care with their neurofibromatosis and support an NF camp. If any of these services can be of great value to your families, please don't hesitate to reach out to me at the NF Network or to Sharon at NF Central Plains. In our advocacy work, as you heard, we need your help. Last February, just before COVID hit, we had almost 100 NF advocates in Washington, DC that were educating Congress on the importance of federal funding for NF research. These results of these precious efforts from the NF advocates is that there now is appropriation committee language to support federal funding of NF research for the year 2021 at the level of $20 million. But we are not done yet. We're going to need your help, your voices, when the time is right to continue to advocate for funding through the Appropriation Committee in order to be able to receive these funds. Every year, the NF research program is not in the president's budget. We must continue to advocate in order to be able to continue receiving this funding every year. You will be receiving emails from us asking you to send a message to your representative and senators, asking them to support the funding of NF through the Appropriations Committee. Without your voices, this funding would not be possible. Please do not hesitate to reach out to Sharon or myself to learn more about this program. The NF Network also educates the NF community about the opportunities for individuals with NF to participate in clinical trials. We have been made aware of an NF1 clinical trial looking at plexiform tumors. By participating in this project, you will help researchers understand the complexity and pain associated with living with plexiform tumors, which may lead to new treatments. You will receive an email early next week from us describing this opportunity. If you have any questions regarding the study, please reach out to us at the NF Network and we would be glad to help you. And now it is my privilege to introduce our first speaker, Dr. Angela Hirbe. She will be presenting on the topic of malignant peripheral nerve-sheath tumors, or MPNSTs, and share her latest research. Dr. Hirbe did much of her training at Washington University in St. Louis and worked in the lab of Dr. David Gutman, whom many of you know. She currently teaches at Washington University, heads her own lab, and treats NF patients with MPNSTs. We are so fortunate to have her with us here today. Dr. Hirbe will leave some time at the end of her presentation to answer your questions. So please leave them in the Q&A box at the bottom of your screen. We will do our very best to get your answers to your questions within this next hour. And now I would like to turn it over to you. Dr. Hirbe, thank you for joining us.
Of course, thank you for having me. All right. It looks like everybody should be able to see my screen at this point. So thank you for the introduction, Kim, and thank you, Sharon and NF Central and the NF Network for the opportunity to talk about NF and some of the research that we're doing today. So the title of my presentation is Clinical Challenges and Emerging Advances in the Diagnosis and Management of Plexiform Neurofibromas and MPNSTs. So just an outline of what I'm going to talk about this morning. So I'll go through a little bit of general background on NF1, how a patient ends up with NF1. And then I will talk a little bit about what are plexiform neurofibromas and MPNST. Then I'll review some of the data for the recently approved drugs for plexiform neurofibromas, which is super exciting that we finally have something that's approved. And then I'll talk about some of the research in my lab and collaborations that we have geared at early diagnosis of not only MPNSTs, but cancers in general in the setting of NF1. And also research geared at finding novel treatments for MPNST, one of the most aggressive cancers that patients with NF1 can potentially face. Just in terms of background, as I know there may be some people in the audience who don't know all of this, although some of you, I may be giving you information you already know. So enjoy the review, I guess. NF1 we know is a clinical syndrome, and patients can have manifestation in many different organ systems. And that is sort of captured in the clinical criteria that we use to make a diagnosis of NF1. So in order to say that somebody has in the axillary or inguinal region, so areas that don't normally see sunlight, or an optic glioma, so those benign tumors that can happen in the optic nerves, the nerves that send to and from the eye. A distinctive osseous or bone lesion, such as sphenoid dysplasia, which is an abnormality in some of the bones in the face, or something called a tibial pseudarthrosis, which is shown in this picture, which basically is the tibia, one of the leg bones, is turned abnormally. And this can cause pain and make it difficult for patients to walk. 2 or more Lish nodules, which are those little benign growths that can happen in patients' eyes. And so those are shown in the picture in the corner. And then obviously a first degree relative with NF1. In terms of things that happen to patients with NF1, these different manifestations, different things that can happen to people with NF1 happen at different times in a patient's lifetime. So things like the cafe au lait macules, JMML, which is a type of leukemia that can happen, plexiform neurofibromas, or the optic pathway tumors in the brain, or the tibial bowing that can happen in the bone. Those things tend to happen early in life. So, you know, they can be there at birth or develop in early childhood in most people. For some people, they'll happen in late childhood. But really, if you make it into adolescence and adulthood and you don't have any of those features, you're not going to end up getting those features. Things like cutaneous neurofibromas tend to start occurring in late childhood, and we see them increase in adolescence and in adulthood. Sometimes they plateau, but they can certainly, in some patients, continue to increase, you know, throughout the lifetime. Things like speech, language, motor, and cognitive manifestations, again, tend to manifest in early to late childhood. And then adolescent phase, and these things have not happened. So now I want to talk about some science basics that I think will be important for just understanding how NF happens, and will be important for kind of understanding the rest of the things that I'm going to talk about. So again, sorry if this is information that some of you already have, but so DNA is the blueprint of life, right? So we have DNA, which is our genetic code, and everybody's code is unique, and that DNA is within every cell in our body. And so, you know, this image is supposed to sort of summarize that for you. And I'm sorry, I'm trying to find the pointer. But we have a person, right, which you can see in the image. And then you see the image of a cell here, right? So all the DNA is housed in the middle of that cell. And you can sort of break the DNA down into more individual pieces. So chromosomes, right? So we have 23 pairs of chromosomes, or 46 chromosomes in every cell in our body. And that DNA, that blueprint, gets turned into RNA and then proteins. And the proteins are really the building blocks in the cells that send all of the messages. And so this very complicated picture sort of shows that. So this is sort of the outside of a cell. And some of these proteins that the body makes, because of that blueprint that it has, make up different transporters and things that are on the surface of the cell. And that's how the cell gets messages from other parts of the body. And then all these other proteins that are made are sending all of these complex messages, basically so that all of your organs and cells can work and our body can function. So NF1 is caused by a change or a mutation in the DNA. So NF1 it's really more than a cancer predisposition syndrome. So yes, people with NF1 are at risk for getting certain cancers, but kind of we talked about in the beginning, it can have different manifestations in the body as well in terms of learning and memory, or the skeleton. So there are a lot of different organ systems that are affected, in addition to putting patients at risk for getting a cancer. And we know this is a very common condition. So about 1 in 2,500 people worldwide have NF1. As I said, it involves numerous organ systems. So the NF1 gene is located on a certain chromosome, remember, we talked about it in the last slide. So this is just a picture of chromosome 17, which is where the NF1 gene sits. And the NF1 gene codes for that protein neurofibromin. So neurofibromin is the protein that does the work. And what does neurofibromin do? So neurofibromin is kind of an important stop sign in our cells. It's one way that the cells know to stop growing. So remember, I told you a couple slides ago when we were sort of explaining how all of this science works. In every cell in the body, you have signals that tell the cell to grow. But in order to keep yourself from getting a tumor, the cells have to know when to stop growing. And so that is one of the things that neurofibromin does. It acts as a stop sign, and it stops these pathways, these other proteins that tell the cell to grow. So because it's such an important stop sign, that is why patients with NF1 are at risk to develop both benign tumors and different cancers. And this table just lists some of those cancers that patients with NF1 are at risk for developing. So optic pathway gliomas, those brain tumors that happen in the nerves that help the eyes to work. High-grade gliomas, those are brain tumors that are more aggressive. MPNST, or malignant peripheral nerve sheath tumor. These are the type of sarcoma or cancer of nerves that patients with NF1 are at risk for. And that is kind of the focus of my research and one of the conditions that I treat in my clinic. Gastrointestinal stromal tumors, or GIST. These are another type of cancer that can happen in the small intestines or the stomach, typically. We know that patients with NF1 are at increased risk for breast cancer. And that's an important thing in terms of screening. It's very important for patients with NF1 to start breast cancer screening earlier than the general population. We usually recommend patients with NF1 start screening with mammograms at age 30 instead of age 40. Leukemias, cancers of the blood cells, are something else that patients with NF1 can be at risk for. Something called a pheochromocytoma, which is a cancer that can happen in the adrenal gland, which is a little gland that sits on top of your kidney and is important for making different hormones. Duodenal carcinoid, so this is another type of cancer that can happen in the small intestine. And then rhabdomyosarcomas, which are another type of sarcoma, but these are cancers that arise in muscle. So now I'll talk a little bit more about MPNSTs and plexiforms. So malignant peripheral nerve sheath tumors are cancers that are arising from the nerves. So they can happen anywhere in the body. And specifically, they arise from something called Schwann cells, which are these cells that actually kind of coat the nerve. And Schwann cells normally help the nerve send messages more quickly. So this is just a picture of what one of these tumors can look like when a surgeon takes it out of the body. And this is what those cells look like under the microscope. So every one of these little purple dots kind of indicates another cell. So lots of cells growing because they don't know when to stop makes this big tumor. And this figure is just meant to show you how an MPNST develops in somebody with NF1. So like we talked about in the beginning, somebody with NF1 has a mutation in one copy of the NF1 gene. So they have one good copy and one good copy and one bad copy. So they still have a little bit of that stop sign. But the problem is if they get a mutation in the other gene, because everybody has two copies of every gene, now you lose that stop sign. And when you lose that stop sign, that's how you can start getting some of the benign tumors that we see in NF1. So like the plexiform neurofibromas. That also happens in the cutaneous neurofibromas, the one on the skin. The difference is these cutaneous neurofibromas on the skin will never become an actual cancer. But these plexiform neurofibromas, the ones that are bigger and involving multiple nerve bundles, are the ones that are at risk for becoming a cancer. In order to become a cancer, though, these tumor cells have to get other mutations in other important genes. So if they start getting mutations in other genes, then they can end up with what we call an atypical neurofibroma. It's kind of an intermediate. It's not quite a cancer. But these atypical neurofibromas, we treat aggressively with surgery. We want to take them out because we know that they are at risk for becoming a cancer if those tumor cells continue to get other mutations in other important genes. And I've listed some of them here. then you end up with a malignant peripheral nerve sheath tumor, this aggressive cancer. We also know that cancer development is very complex. So not only, you remember all those signaling pathways that I showed you going on inside the cancer cell, but also the other cells in your body can affect the growth of the tumor as well. So your normal blood cells and the other kind of supporting cells in the environment can send signals that affect the growth of the tumor. And this is also more complex in patients with NF1 because I say normal cells, but remember that all of these other cells in the body that are important, they also have lost one copy of the NF1 gene. And so the signals that these cells send are probably different than in somebody who doesn't have NF1. And that's something that we still need a lot more research to fully understand. So I said that plexiform neurofibromas are at risk for becoming MPNSTs. And about a third of patients with NF1 will get these plexiform neurofibromas. And I'm showing one here. So these kind of big tumors that are multiple nerve bundles are affected. About 10% of patients with NF1 ultimately will develop malignant peripheral nerve sheath tumors. So not everybody, but certainly a high enough number that we would like to do a better job of being able to figure out who is at risk for getting these cancers and find a better way to diagnose them early. So these are what I think are some of the very important clinical challenges that we have. How should we treat plexiform neurofibromas? The problem is that surgery is not so easy, right? This is a very involved tumor. It's right by the spine. Others take up, an entire arm or an entire leg. So there's no way to really remove the entire tumor. And if you leave any of the cells behind, they're just going to start growing back. And the other thing is the entire tumor may not become a cancer. It may be that a cancer will develop in a specific part of the tumor and only that part needs to be removed. So how should we monitor these plexiform neurofibromas so that we can catch an MPNST if it happens early? And then what's the best way to treat an MPNST? So first, let's touch on plexiform neurofibromas. So remember, you lose the other copy of the NF1 gene, and I'm sorry, my pointer doesn't want to behave this morning. You lose that stop sign fully, and you end up with this benign tumor. And we know that some of the signals from the other cells in the body are kind of feeding in and helping that tumor grow. So a lot of this work, research came from Wade Clapp's group in Indiana. And there is a drug called imatinib or Gleevec, and it can inhibit some of those cells in the body that we know are important for sending signals to these plexiform neurofibromas. And so what they did here is they treated some cells in a petri dish in the lab with this drug imatinib. And you can see if they increase the concentration of the drug, you have less live plexiform tumor cells. So no drug, 100% of the cells are alive. A little bit of the drug, 75% are alive. A little more of the drug, 70%, and a little more of the drug, 50%. So then, this drug was taken into a clinical trial. So with patients, and this drug was already approved for treating other cancer conditions. So it was known that this was a safe drug. But would it be effective at treating plexiform neurofibromas in people? And so these graphs are from that clinical trial. And this top graph is called a spider plot. So you can see every patient is indicated by a separate line. So these red lines were showing patients that were treated with the drug, but despite being treated with the drug, their tumor still grew. So when the lines are going up, it means the tumor is growing. If the line's going straight across, it means the tumor is stable. These lines where they're going down are patients whose tumors shrink when they were on the medication. And then similarly, you can see that in what we call a waterfall plot below. So if the bar is going up, it means that the tumor is growing. And if the bar is going down, which are the green bars, you can see that the tumors are shrinking. But in the best case scenario, they only had about a 20% shrinkage in the tumor, and certainly not in a high percentage of patients. So it doesn't seem that this strategy is something that's really going to be beneficial for shrinking these plexiform neurofibromas. But what has been even more exciting, and this is very recent, is that we now have the drug selumetinib, which has been studied for several years now, which is approved for treating these plexiform neurofibromas. So again, I still have my little diagram up here reminding you NF1's our stop sign. You lose it, you get this plexiform neurofibroma. What does selumetinib do? So remember, in patients with NF1, I X'd out neurofibromin in this image because you lost neurofibromin, which is normally a stop sign. So selumetinib is a drug that can act as that stop sign. So the thing that neurofibromin tells to stop is this protein MEK, because MEK tells the cells to grow. So selumetinib is something called a MEK inhibitor. So it blocks this protein MEK, and so it's acting as a stop sign, so kind of replacing neurofibromin. The thing is, neurofibromin affects other pathways as well. So there's still so much research that's needed, because it may be that a MEK inhibitor alone is not enough, because you're putting kind of one stop sign back in, but some of these other pathways have proteins that can still be active. And so again, we're still trying to understand how all of those pathways work together. This is a waterfall plot, again, like I showed you in the last slide, but you can see here, a lot more exciting than the last one. We don't see the bars going up. We see all the bars going down. You know, some of the patients didn't have very much change in the tumor. I mean, this is less than 10% change, so we don't necessarily think that that's clinically significant. But some of these tumors shrank by almost 50%, which is pretty amazing. And this is an image that just sort of shows you some of the MRIs from a patient baseline after cycle 5, after cycle 10, and then kind of pictures of the patient. And you can see how that tumor shrinks over time, and how sort of abnormalities that you can see in the patient's stance, and how you see that tumor has improved over time as well. There are still so many unanswered questions, and somebody emailed some of these questions yesterday. I already had this slide in here, but you're right. We don't know how long somebody should be on a MEK inhibitor. The trials were for 2 years. Some patients continue on if they had a good response, but we don't know if it should be lifelong. We don't know if 2 years is enough. We don't know which MEK inhibitor is best. So selumetinib is FDA approved. SpringWorks has a trial right now with binimetinib. We have that trial open at WashU, and I know it's open at a number of other sites. Trametinib is another MEK inhibitor, and there may be others that are being developed. And then the question that I sort of brought up in the last slide is, are there other pathways that we should target along with the MEK inhibitor? So is there a combination therapy that would be better? We don't know. These are all super important questions to answer, which is why we still need a lot more research for NF1. So now I'm going to talk a little bit specifically about what my lab is doing. So I think our patients are awesome, and they want to help make progress in this condition. And so I don't think I've ever had a patient kind of decline signing up for any of our research clinical trials, where basically our patients give us permission to take tumor samples, so extra tumor, you know, that was removed because of surgeries that they had or biopsies that they had, and take some of that tumor that's left over into the lab to study it. And so we sequence the tumors, so we find out exactly what the DNA is in these tumors compared to what normal DNA should look like, right? So we figure out what are all the changes in all of these different patients' tumors. And then we take our models that we have in the lab, which are sometimes cells that we grow in a petri dish, sometimes they are mouse models. And we try to see if we can use what we learned from the genetics from the tumor samples to better understand ways that we can diagnose these tumors earlier and potentially ways that we can treat these tumors better. So in terms of diagnosis, the current imaging that we have is challenging. So MRI, which is what's shown here, is not perfect for us trying to figure out, is this plexiform still benign or is it becoming a cancer? There's something called PET scans, which some of you may have heard of. So a PET scan works by basically injecting a labeled sugar molecule. So all of our cells use sugar. Cancer cells tend to use sugar more and like sugar more. And so if somebody has cancer, often they take up this labeled sugar, and then on a scan, it lights up bright, like you can see in this image here. That's not perfect either for malignant peripheral nerve sheath tumors. So sometimes these plexiforms light up really bright and they're not a cancer. And sometimes the cancers don't light up and they are a cancer. So we really need a better test in order to know when somebody's developing a cancer. So there are other proteins on the cells, and some of them are labeled here, right? So all these, what we call transporters on the cells, are the ways that cells get in the building blocks that they need. So the sugars and the little amino acids, which make proteins, those are all kind of breakdown products from the food that you eat. And those have to get into the cell somehow for a cell to continue to get energy and work. Transporters are also the way that we get drugs into cells. So, you know, if somebody's treated with chemotherapy, for example, the chemotherapy goes in through certain transporters and gets into the cell and, you know, kills cancer cells. So our lab found that this particular protein transporter, ASCT2, seems to be on the malignant peripheral nerve sheath tumor cells. And there is another imaging compound, something called fluciclovine, that could potentially be used to image these tumors. And so that's something that our lab is looking at. So here you can see this is checking to see if this certain protein is in a certain cell type. So in Schwann cells, normal nerve cells, we don't see this protein. But in these malignant peripheral nerve sheath tumor cells, we do find this protein. And when we looked at some plexiform neurofibromas from patients, we did not see this protein. But again, we do see it in malignant peripheral nerve sheath tumors. And so one of the things that we're looking at in the lab now is if we can use this particular imaging compound, this labeled fluciclovine, which goes into a cell through this protein ASCT2. So can we use this to help us distinguish MPNSTs from plexiform neurofibromas? And this is a project that we've just started in the lab. So I don't have any other data to show you, but this could potentially be an exciting way to better diagnose these tumors. One of the other things that we're looking into is trying to see if we can detect whether or not a plexiform neurofibroma is becoming a cancer just by a simple blood test. So what you can see here are supposed to be pictures of these nasty little tumor cells, which again are filled with DNA, just like you have in every normal cell in your body as well. But over time, both normal cells and tumor cells will die. It's just that tumor cells keep growing so fast that they replenish themselves faster than they die off, which is why tumors continue to grow. But when these cells die, some of their DNA goes into the bloodstream. So this is supposed to represent a blood vessel here. So you get what we call cell-free DNA. So it's not the cells that are in the blood, but just the DNA from the cells. And so you can have normal cell-free DNA. So from some of your normal cells, like these cells surrounding the blood vessels, over time, when one cell dies and a new one is formed, that normal cell-free DNA ends up in the blood. But then also this cell tumor or ctDNA, as the tumor cells die off, that ctDNA gets into the blood. And so what we want to do is see if we can take blood samples where we get that cell tumor DNA, and can we use it to try to not only diagnose a cancer earlier, so using it as a screening test, but can we use it to detect if there's any residual disease left after a patient has had surgery or some other treatment? And can we also use it to monitor treatment response? So can we use this instead of imaging, just a simple blood test, and see, is this treatment working? Are the levels of that ctDNA, the cell tumor DNA, going down, or are they going up? In which case we would know that the treatment isn't working. And so this work we've been doing in collaboration with Adil Chowdhury's lab, who's another researcher at WashU. He's a radiation oncologist. And then Jack Schern, who is a physician scientist at the National Cancer Institute, works very closely with Birgitta Wiedemann. And so Birgitta's also been supplying samples for this study. And so I'm showing the pictures of some of the people in the lab who are doing this work. Paul Jones, who's a graduate student, Jeff Szymanski, who is a bioinformatics specialist, Divyas Rahari, who's an undergraduate in the lab, and then Taylor Sundby is a pediatric oncology fellow at Johns Hopkins and the National Cancer Institute, and Taylor works in Jack's lab. So we have taken samples, blood samples from 43 patients with plexiform neurofibromas and 12 patients with malignant peripheral nerve sheath tumors. And from some of them, we also have some of the DNA from the tumor to compare. But basically, we isolate that cell tumor DNA from the blood samples. And then we do DNA sequencing. So we figure out exactly what the sequence is in that DNA. So what is the blueprint in this tumor compared to another one? And can we then use that sequence from the plexiform neurofibroma and the MPNST to distinguish them? And one of the ways that we knew that this could be possible is because if we looked at the actual tumors from the patient, so if we take a tumor out of a patient with a plexiform neurofibroma, we don't see nearly as many changes. So what happens with malignant peripheral nerve sheath tumors is they start getting extra copies of the genes and such, and they lose copies of other genes. And so what these figures are basically showing you is every time a gene or chromosome area is lost, it's in blue. And when something is gained, when they get extra copies, it's in red. And white means that it's normal. So the normal state of having just 2 copies of every gene. So plexiform neurofibromas are mostly white. So mostly just normal numbers of chromosomes and genes. But in MPNSTs, they're mostly blue and red. So you're getting lots of changes. And so the exciting thing is that we're able to see this with just a blood sample as well, not from getting the whole tumor and sequencing it, but just from taking a blood sample from a patient and then getting that DNA and sequencing it. We can see this is from a patient with a plexiform neurofibroma, just this straight line across, meaning that they just have 2 copies of every chromosome area. Whereas in this patient with a malignant peripheral nerve sheath tumor, we start getting these areas of gains and losses. So these ups and downs. And so we're able to tell that this blood sample is from a patient with a malignant peripheral nerve sheath tumor, and this patient just has a plexiform neurofibroma. And so from that whole data set that I showed you, we got some blood from healthy people too. You can see plexiform neurofibromas fall around healthies, and we see much higher levels in these patients with malignant peripheral nerve sheath tumors. And so we're able to distinguish this with a pretty high specificity, and we're still working on better ways to refine this test to make it even more sensitive and specific. But this is the first time this has been done for patients with NF1, and very exciting. And we have so much more work planned in this regard, but we're hoping to publish this data soon, and then are looking to try to get funding to really further develop this, because I think it would be great for patients with NF1. As I mentioned, we also want to see if we can use this to track treatment responses. And so you can see here, these are some CAT scans from one of our patients that has a malignant peripheral nerve sheath tumor and was going through chemotherapy. And so you can see that unfortunately this tumor here grew on one type of therapy, but then when we changed therapy, we can see that the tumor started to shrink again. And so these are the measurements from imaging in blue. So we can see, unfortunately, the tumor is growing, but then when we switch therapies, we can see that the tumor was shrinking. And we see the same thing in the bloodstream. So, you know, when there was pretty much no cancer on the imaging, that's what we saw in the blood. When the tumor started to grow, we detect that by the blood samples that we took. And when the patient started to respond to chemotherapy, we're able to see that just by checking in the blood and not necessarily by doing this imaging test. And then the question becomes also, can we use this to detect recurrence? So when you've taken a cancer out completely and then you're following the patient by imaging to see, is this cancer going to come back or not? You know, can we use a blood test to do that? And so these are images from another patient who had, you can see here, their tumor after surgery was removed. So we don't see any tumor on the scan. So in blue here, we see no tumor on the scan. It was about a week before that we got a blood sample. But again, we don't detect that cancer in the bloodstream. Here, when the patient had a follow-up scan, we still didn't see any cancer by imaging. We don't see it in this CAT scan, but we detected something in the bloodstream. And so we're worried that, you know, potentially the cancer could be coming back. And then you can see here at this later follow-up date, indeed, now we can see something on imaging, the cancer was back. Again, this is research. This blood test isn't being done as a clinical test because we don't fully know our sensitivity and specificity yet, nor how to interpret everything. So we have subsequent blood samples on the patient, but these haven't been analyzed yet. But this is, again, why we need more research so we can figure out exactly you know, whether or not we can trust this in every patient situation. This is just one patient, but in order for something to become a test that's used, we have to prove that it works, you know, for most patients, if not all patients. Now, what do we do? How do we treat an MPNST once a patient is diagnosed? So what we've been talking about now is how can we diagnose these early? But then what happens when we do get an MPNST? So I do think it is very important for anyone with NF1 who ends up getting a cancer to really be seen at a center that has a lot of expertise in caring for patients with NF1 and in caring for sarcomas. At our site, as at many academic sites, we take a very multidisciplinary approach. And so any patient gets their case presented at our tumor board that involves the surgeons, the radiation oncologist, and the medical oncologist like myself. And for these malignant peripheral nerve sheath tumors, they are one of the most aggressive types of sarcomas. And so we do tend to treat them with surgery, radiation, and chemotherapy. One of the things that has been sort of debated over time is whether or not MPNSTs really respond to chemotherapy. And so this was just a small study that we did where we looked at patients treated at our site who had tumors that were too big initially to remove by surgery. And so in that setting, we started with chemotherapy. One of the advantages to starting with chemotherapy is we can see if the tumor actually responds or not. And so this table is just showing that there were 5 patients that we looked at. Most of them were in their 20s to 30s. There were 2 patients in their 50s. These tumors were in different places. Some of the patients had NF1. Some of the patients just had sporadic malignant peripheral nerve sheath tumors. They were all high-grade tumors. This lists the number of cycles of chemotherapy that they got. And what you can see is that 4 of the patients had decrease in their tumors by a pretty significant amount. And even the one that the tumor didn't shrink, when we took the tumor out by surgery, we could see that there was some response of the chemotherapy. There were some dead cells that were in that specimen. And so this is just, again, graphs from the imaging. For every individual patient here, we can see that the tumors did decrease. In one patient, it just stayed stable. So again, you can see these waterfall plots, shrinkage of the tumors. This is just a scan showing you a lymph node area in one of the patients. So this bigger tumor here shrank down to this tiny spot. And then there were a subset of patients that had PET scans also. That's where they get that labeled sugar. So the cancers light up bright. And typically, if a cancer is responding, we'll see that brightness decrease on subsequent scans. One of the patients actually had a complete response by the PET scan. So, you know, they went from having this high bright signal to no signal. So for patients that have MPNSTs that are just in one spot where it hasn't spread, where we've caught it early enough, those patients we typically treat with surgery and radiation and chemotherapy to try to really reduce the risk of that cancer coming back. Unfortunately, some patients have their cancer come back and it spreads to other places, or some patients don't have their cancer caught early. And so when it's caught, it's already spread. And when these cancers spread, they tend to go to the lungs or to the bones. So again, in this setting, we really don't know the right way to treat these patients. And this is where clinical trials become very important so that we can find better therapies that work. And so I do think it's very important for patients with MPNSTs that have spread to really be seen at these NF sarcoma centers and to really consider a clinical trial, because there are some chemotherapy drugs that we can use, but we know that they're not going to cure patients and they're going to buy them a limited amount of time. These are a lot of the clinical trials that have been done before. And unfortunately, none of these have ended up leading to any successful therapy that's become approved. There are 3 clinical trials that I am aware of that are open right now. This pexidartinib, sirolimus. So sirolimus is a drug that hits the mTOR pathway, one of these pathways that we know is active in NF. Pexidartinib is a drug that's actually hitting we call the microenvironment, those supporting cells, kind of similar to the imatinib that we talked about earlier. So this combination is in clinical trials, and we have the study open at WashU, and it's also open at Columbia, which is the site in New York that started this study. This selumetinib sirolimus, so selumetinib is that MEK inhibitor that we talked about earlier. And again, sirolimus hits the mTOR pathway. So you're hitting 2 different pathways that we know are active in these cancer cells. And this is a trial that's being run by the NF Trials Consortium and SARC, the SARC Alliance. And so it's open at numerous sites, including the NCI, Johns Hopkins, and WashU. This is a newer trial that opened recently. And this, I think, is a very exciting trial. So this is one drug that is an MDM2 inhibitor. So again, hitting another pathway that tells cells to grow. And then a drug called pembrolizumab, which is one of these newer drugs. People may have seen commercials for this. So Keytruda is the other name for it. But this drug class is something called immunotherapy. So these drugs don't hit the tumor cells. What they do is they help to reactivate your own immune cells so that your immune system then attacks the cancer. So this has been a really exciting avenue of research in a lot of other cancers. These immunotherapy drugs have really, changed the prognosis in a good way for patients with melanomas and also lung cancer. And we're really just starting to look at these drugs in sarcomas and in patients with NF1. So these are some of the trials that are open right now. My lab is also working together with a couple other labs to try to develop better therapies for these tumors and to come up with other good ideas for clinical trials. And so I have a collaborative grant with Christine Pertilis' group at Johns Hopkins University, and work closely with NTAP there. And then also with David Largaspada at the University of Minnesota. And so what our 3 sites are doing are to try to develop a better model that really encompasses all of the differences and heterogeneity that we can see among different patients by developing what we call patient-derived xenograft models. So when our patients go to surgery or get a biopsy, we take a piece of that tumor and it gets implanted into a mouse. And then it gets passage, so implant it serially in mice so that we can do these drug testing experiments, in an actual environment more similar to the human, instead of just looking at cells growing on a petri dish. And so what we hope to do is basically continue to generate these lines, and basically we're sequencing all of them so we know what genetic changes are in all of these different tumors. And Dr. Sarah Goslin at Pacific Northwest National Laboratory is kind of helping us with that sequencing bioinformatics piece. And then David Largaspada's group in Minnesota is doing kind of a drug screening in the lab, not in the mice, and I'm going to show you a picture of what that looks like in a minute, because it's also not just growing these cells on plastic. And then again, we're trying to see if we can come up with predictions. So do certain genetic changes in the tumor predict a response to a certain drug cocktail? And then Christine's group at Johns Hopkins and my group, my lab, will then test the best combinations in the actual mice. And then we hope that is going to lead to a better clinical trial where we sort of pick what drugs based on the genetic changes in a given tumor. And so again, we're trying to build this infrastructure so that we can look at how the tumor grows, look at the genetics, and then figure out what correlates there are so we can do these predictive modeling. And so the sort of whole pipeline that we're using is basically doing some prick screening just with those cell lines that we have grown on plastic, just so we can get an idea of what doses of the drugs. And then Dr. Largaspada's lab is really using this 3-dimensional drug screen. So instead of the cells just growing on plastic, they're grown in these little microseals with collagen, and we can actually add some of the other cells that are part of the tumor microenvironment and other components that are normally in the matrix around the tumor cells. And so, a lot of times you can kill cells really easily on plastic, but it's not that easy in an actual person or mouse, right? And so this will make it a little better for us to predict what's going to work in an actual, mouse or human, because it's a little harder to kill those cells when they're in this 3-dimensional matrix with some of the other cells and components that help them to grow. And so we think that this is a really key piece to kind of coming up with better drug combinations. So, in conclusion, I think NF is a very complex diagnosis, and it affects multiple systems in the patient, and it also puts them at risk for developing both benign tumors and cancers. I think it's very important for patients with NF1 to be cared for at a multidisciplinary center with expertise in NF1. And if someone does develop a malignant peripheral nerve sheath tumor, to be seen at a center that also has expertise in caring for sarcomas. Very exciting that we now have MEK inhibitors to treat plexiform neurofibromas, but there are still many more unanswered questions that we have in terms of how to best use those drugs. And future work in my lab is really aimed at trying to help with early cancer detection so that if we find an MPNST, we find it when it's small and we can get it out and hopefully never have to worry about it. But we're still continuing to look at improving therapies for both plexiform neurofibromas and also for malignant peripheral nerve tumors that have spread. Because there will still unfortunately be situations where patients aren't diagnosed until their cancer has already spread. So I just want to acknowledge everybody that's helped with this work. This is a picture of my lab in the virtual setting now since you know, everyone is kind of in the lab on shifts so we can social distance and keep our space, and our lab meetings are all by Zoom. And so everybody in my lab and kind of former members that have contributed to this work, the Chowdhury lab and the Shern lab, who have been you know, very important in this early cancer detection piece. And then the preclinical drug testing and the patient-derived xenograft work is certainly a collaboration with Johns Hopkins University, the University of Minnesota, and Dr. Goslin at PNNL. And then Dr. Alloway at Sage Bionetworks is helpful in terms of helping us upload all of the data so it can all be shared. We are very much in favor of openly sharing data and trying to foster collaboration and working together, because I think that is so key to helping us make progress. And so all of our data is uploaded for that NF Open Network Initiative. And then I've listed our funders here. Obviously, we could not do research without the funding, and so that's a really important piece to the advocacy that you all help with. And then my lab website is here in case anybody has any, you know, interest in kind of looking further at what's going on. And so I'll stop for questions. These are my three crazy kiddos who are locked in the backyard right now, and I will spend time with when this talk is over.
Perfect. Thank you so much, Angela. It looks like we do have quite a few questions in the Q&A box. Did you want to go ahead and take a look at those?
Yes, I just stopped screen sharing so that I can see it, because I don't think I can see it otherwise. So let's go to, let's see. So will there be research for mosaic NF so we have more information and care for these kids and don't have to fight so hard for these kids in their care? A very good question. Certainly, that's not something that my lab is kind of actively working on, but you know, I still think that we can learn things from the models that we have and apply that to patients who are mosaic for NF1. So in these patients, it's not that genetic change happened at the very beginning in embryonic development. It happens later. And so you don't have that loss of NF1 in every cell in the body. And so screening, certainly I think the screening that we're doing would still be applicable to those patients with mosaicism. And even treatment paradigms that we look at would still be applicable in that situation. But you're right, we don't have a good model of that to study that setting perfectly, but you know, that is something that researchers are interested in. Going on down here, let's see. Someone asked, what causes a plexiform neurofibroma? So I guess you're asking the question, why do some patients get a plexiform neurofibroma and others don't? And we don't have a good answer to that. You know, there have to be other things, whether it's in the environment around us or other changes in the DNA and the blueprint, you know, in other genes that maybe put some patients at risk for getting plexiform neurofibromas while others don't. But certainly, you know, there are other labs who are really focused on trying to predict why certain patients get certain tumors and others don't. I mean, what we can tell you is sort of what I showed in the slides, that if a patient loses that other copy of NF1 in a Schwann, that's how you get a plexiform neurofibroma. But why did the patient lose that other copy of the NF1 gene? And that I don't know how to answer at this time. That is something that we're still trying to figure out. How is a GIST diagnosed? If you have an MPNST in your abdomen, or you're more likely to get a GIST. These are kind of jumping around. So how is a GIST diagnosed? So right now, I mean, sometimes on the imaging, the radiologist can see the way a tumor looks, and they may say that this is probably a GIST based on the way it looks. But the only way that we have to know right now is actually to do a biopsy and actually look at the cells under the microscope to see what they are. Now, the genetic changes that you have in a GIST are different from the genetic changes that you have in a plexiform neurofibroma or an MPNST. So that may be something that we can ultimately use our liquid biopsy assay to figure out, but certainly we would need a lot more research in order to do that. But right now, basically, it's by biopsy. If you had an MPNST in your abdomen, are you more likely to get a GIST? Not that we know of. No, there's no necessarily association between GIST and MPNST. Somebody wrote, Dr. Alan Bellsberg, based out of Baltimore. I know Dr. Bellsberg. Chose to take my son's case and did surgery ASAP because there was a cell he had inside the tumor near the peripheral nerve. What are the chances of it becoming a cancer? That's a hard question for me to answer. Again, it's more of like a specific medical question because I didn't see any of the imaging or, you know, the pathology that goes along with that. So that's hard to say. But my guess is that maybe what he saw was what we call that atypical neurofibroma. Those atypical cells, if you remember that slide that I showed where you see a plexiform goes to an atypical, goes to an MPNST. And so often we say if we see those changes indicative of an atypical neurofibroma, we're going to take that out because we think that there's a chance that it can become an MPNST. But again, I don't know in this specific situation. Can you segment those pictures by age and sex? I don't know what picture we're referring to on this one. So if somebody wants to kind of email further question on what they were asking, I would be happy to try to address that. And then someone, maybe the green is for a particular cluster. Oh, question was for imatinib. So if you were talking about the imatinib slide, I think you were talking about the different graphs that we had. So basically, every line was meant to represent the growth of a different patient's tumor. So the red lines that were going up were meaning that the tumor was growing, and the green lines going down were meaning that the tumor was shrinking. So they were just showing the patients that responded with green lines and the patient whose tumors didn't respond with red lines that were going up. Can you segment the response by sex and age? Oh, I guess that was for imatinib. I'm sure they looked at that. I don't have those graphs pulled in the actual papers, but there was no difference in response by gender. So gender didn't matter in terms of response. And as far as age, most of the patients in those studies were pediatric patients. They were children. I don't have the exact age breakdown, but again, in those studies, it was pediatric, so patients under the age of 18. How would you characterize the burnout for a cancer drug? For NF2, I know a patient reporting having to move from one drug to the next. Would you say that this is a result of more resistant tumor cells surviving earlier treatments? Yes. So cancer cells are very tricky. So if you can't kill every single cell, then ultimately the cells that are left change their DNA, change their blueprints, and find a way to resist whatever treatment they're doing. And that is a big problem that we have. That's also something that we're trying to look at in the research that I'm doing with Dr. Perchilis and Dr. Largaspada. In the studies that we're doing, when we treat these tumor cells over time, we are collecting some of the DNA and the protein at different time points, including when those cells develop resistance. So, you know, when we're doing research in the lab, like we may treat these tumors in the mice with drugs, and we may see shrinkage for a while. And then at some point, that tumor cell starts growing. And so we're trying to collect the tumors at that time and then look at what changes have happened in that tumor cell at that point, so we can try to figure out what some of these resistance pathways are, so that we can refine our treatments to try to develop cocktails, multi-drug combinations that could hopefully prevent that from happening. Can I donate cells taken from my surgery to remove an MPNST? How could I do that? So if you want to reach out to me specifically, which you can do through the lab website, that may be able to done through the NIH. I think that they have a protocol where they can consent patients by phone. I am not allowed to do that at WashU. So typically, you know, at academic centers, a lot of researchers will have a protocol that's approved by our ethics boards to make sure that we're doing everything we can to protect patients' privacy and such. So I have such a protocol at Washington University, so I can offer, you know, that research protocol to my patients that I see, and we can collect their tumors to study in the lab. Problem is, if somebody is at some other site, you know, around the country, I don't have any kind of protocol that gives me permission to get consent from patients over the phone and then request that tissue. But I do think that the National Cancer Institute has such a protocol that Dr. Wiedemann runs. So if you want to reach out to me by email or my lab website, then I can try to connect you in that regard.
Dr. Hirbe, I think we've got time for about one more question.
Okay. In terms of MRI, one patient reported getting much different readings from an NF clinic and a local non-NF radiologist. Can you characterize typical mistakes of a non-F? No, I can't. It's really just a matter, that's why I say it's very important to see somebody at an NF center, because it's just having that experience over time that really helps you. So I think that's unfortunately why we see late diagnoses and things, you know, if patients are in a smaller site where they just don't see that volume. So I can, if Brianna, if you want to email me the rest of these questions, I can try to type up some answers and send them back out. There are a lot of great questions that people.
Absolutely, yes. Thank you so much, Dr. Hirbe, for putting on such an incredible presentation and taking the time to answer those questions. For those of you whose questions we did not get to, we will be sure to get back in touch with you. I will be sure to get those to Dr. Hirbe. Okay. Once again, thank you so much for putting this on and putting together such a great presentation. It was so insightful, and thank you for being a part of our day of information today. Our next presentation, Breakthroughs in NF2 Research with John and Linda Manth, will begin at 10:30. So please, if you haven't registered, make sure you do, and we look forward to seeing you there and having you join us throughout the day. Thank you so much.
Bye.